Is an open access paper which all of you can have access through this PubMed identificator, and this is the title. So briefly, before start discussing the paper, I would like to recap what DNA methylation profiling is. So the methylation is the introduction of a methyl group in the fifth position in the cytosine ring, forming the 5-methyl cytosine. So this process is catalyzed by methyltransferases. And the methylation occurs mainly where the cytosine is followed by a guanine in the CpG islands. So we have several CpG islands methylated in our genome, and it makes a signature, a methylation profile for each cell type and also for many of the tumor types. And the methylation is also important for oncogenesis. If we have a global DNA hypomethylation, for example, in diffuse midline glioma, H3K27M mutant, we have this hypomethylation, and anytime you performs hypomethylation of a promoter region of a gene, that gene becomes overexpressed. On the other side, if you hypermethylate the promoter region of a gene, that gene is silenced, is not expressed. So whenever you have global DNA hypomethylation, several oncogenes will be expressed, and it will lead to DNA mutations and genome instability, and the cell goes into the cell cycle and undergoes cell proliferation and cancer development and progression. Also, if we have a methylation of a suppressor gene, that gene is not expressed, and then we have cancer promotion. And also, if we have hypomethylation of a proto-oncogene, that gene will be expressed, and everything will lead to cancer development.